In section 6.5, you're going to explain what determines blood types in humans. Now, I'm going to give you a lot of background on blood typing. In lab, you're also going to be talking about blood typing. So between lecture and lab, I'm hoping that you understand this material. Blood typing can be challenging for some folks. So if you're having a problem with blood typing and you need to talk about it a little bit more, email me and we can set up a time to talk about it. I'm glad to help out. So, meanwhile, explain what determines blood types in humans. So, we've all heard of blood type, and a lot of us even know our blood type. And it turns out that blood type is determined by the proteins on the surface of our red blood cells. And so, remember, we said when we looked at bacteria, when we were looking at antibodies and talking about making antibodies, we said bacteria have proteins on their surfaces called antigens. And that's how our body knows that the bacteria are foreign because it can read those antigens. Well, red blood cells have antigens or proteins on their surfaces too, and those are proteins that tell our bodies that they're our own cells, so don't kill them. So all cells have proteins on their surfaces, also called antigens. A blood transfusion is when you transfer blood from one person to another. And there's all sorts of reasons why we would need blood transfusions, all sorts of medical reasons where somebody has an issue that they might need a blood transfusion. So when you do a blood transfusion, you have to match blood types. It says to ensure blood types are compatible, you have to prevent agglutination or clumping of red blood cells. And so when you do a blood transfusion, you have to match blood types, or else the blood actually can clump inside of the person and cause very serious, potentially fatal consequences. Okay, so how do we know what the blood types are? So here's all the possible blood types. A negative, A positive. B negative, B positive, AB negative, AB positive, O negative, O positive. So there's two kind of things going on here. We have the A, B, and O blood typing, and then we have this positive negative thing going on. So if we just start with the A, B, and O, and then we'll add the positive and negative on after that. So what this means is, so here's all the negatives up here. So we have A, B, and O antigens. There's three different proteins that are used primarily for blood typing on human blood cells. And they're proteins called A. And so in this diagram, the A proteins or the A antigens are these yellow triangles. The B proteins or the B antigens are the blue circles. And O actually means zero. It means it has no proteins on the surface. So just ignore the negative and the positive right now. So as far as A, B, O blood typing, there's four types of blood. You can have A antigens. We would say you have type A. You have B antigens. We say you have type B. You could have A and B antigens, so you have type AB blood, or you don't have any proteins, any AB proteins on the surface, and we would say you have type O blood. Okay, so these are the antigens. These are the molecules that determine if you have type A, B, AB, or O. Now, if we add in a third antigen, it's called RH. And sometimes, you see it called D also. But RH actually stands for rhesus, which is the kind of monkey that it was discovered in. If you have RH, and so if you look at this picture, the RHs are these little black things. If you have RH, it means you're positive. If you don't have RH, you're negative. So A positive blood has A proteins and RH. Remember, the RH is what makes it positive. RH negative blood has A negative blood has A proteins And that's it. Negative means there's no Rh proteins. Type B positive means you have B proteins and you have Rh proteins. Rh because it's positive. B negative blood means you just have B proteins. It's negative because there's no RHs. AB positive, you have A proteins, you have B proteins, and the Rh proteins make it positive. AB negative, you just have A proteins and B proteins. So there's actually eight blood types for humans. Okay, so the proteins on the outside, we say those are the blood antigens, whether you have A, B, or H. Remember, O doesn't mean you have O antigens. It means you have you have neither A nor B. Positive and negative. Positive means you have Rh. Negative means you don't have Rh. So these are just proteins on the surface of the cell, identifying what kind of cells these are. There's another little trick that's happening. So if a person has type A blood, they can make antibodies. Remember we said cells, some of the white blood cells make antibodies to fight off things that are foreign. So a person with type A blood would find type B blood to be foreign. These B antigens are foreign to this person with A, just like a bacterial cell would be foreign to this person. So they can make antibodies to fight off the Bs. So this would be why somebody with type A blood, if you transfused them with type B blood, their blood would fight off the Bs with their own antibodies. And that would cause agglutination, and that could be dangerous for the patient receiving the blood. Somebody with type B blood would fight off A's. A's would be foreign. So they're going to make antibodies against the A's, and they're going to fight off blood cells that have A antigens. Now, somebody who has type AB blood, they have A antigens and they have B antigens. So they don't make A or B antibodies because then they'd be fighting off themselves. You don't want to fight off yourself. So they don't make A or B antibodies. And then somebody with type O, both A and B are foreign to them. So they're going to fight off any blood cells that have A or B. They're going to make A and B antibodies. If you're just interested in the incidence here, you can see that in the U.S. for Caucasian people, type O and type A are the most common. B and AB are less common. And because this is a genetic factor, this can vary based on your genetic background. So African Americans have a little bit difference in distribution. Native Americans, look at that. Most Native Americans have type O and not these other types. Anyway. Okay, so how do we know what kind of blood you can give somebody? So it says individuals have antibodies against the antigens not on their own cells. If you give them something foreign, they're going to fight it off. They're going to make antibodies against it. If the recipient of a blood transfusion has antibodies against the donated cells, a severe and possibly fatal reaction can occur. So again, if we go back here, if somebody has type A blood and you give them B antigens, blood with B antigens on it, their body is going to fight that B blood away. If somebody with type O blood, if you give them type A, they're going to make antibodies against it and they're going to fight that blood. And so that's a potentially fatal reaction. You can't do that safely. So what happens when antibodies and blood cells get together. Well, there's some blood cells, and oh, there's a foreign cell. This person made some antibodies. Here's a B blood cell. Their antibodies are going to stick to that. And if you have a bunch of antibodies and a bunch of cells, you can see that it can kind of clump up together. And we call that agglutinating, when the cells and the antibodies clump together. And again, that can be, if that's happening inside of somebody's body, that can be a serious problem. All right, so hopefully I've convinced you that there's 8 different blood types, 3 different antigens, and when we do blood transfusions, you can only give people blood that they're compatible with. So let's just look at what kind of blood somebody can receive. If we look at this person, let's start with this person that's an A negative blood. The rule is you can only give this person blood that you can't give them anything foreign. So if I gave this, I have this A person, if I gave them B blood, these B proteins would be foreign. So you would fight off that blood. So that's not compatible. Look at this AB blood. If I took AB blood and I gave it to the A person, well, they have A's, so that's good. But these B's will still be foreign to this person. This person cannot take any blood with B's on it. They can't take that blood. Can this A negative blood receive O negative blood? Let's see, if I give this O blood to this A negative person, I'm not giving them anything foreign. There's no proteins on that blood that this can react to. So yes, I can give O negative to an A negative person. Can I give A positive to A negative? So this person has A negative, this has A positive. If I give A positive to an A negative person, the A's are complementary, that's okay. But this person has these Rh proteins, remember? And they will be foreign to this A negative person. So the A negative person cannot take A positive blood. But let's try the reverse. Can the A positive person take A negative blood? Let's see. Am I giving this person anything new? Well, they already have A proteins, so they can take those. And yeah, this A positive can receive A negative safely. So we're going to do a little bit more of that in lab and kind of practice what kind of blood you can donate and receive and just talk about that a little bit more. But what you'll notice is there's one kind of blood that you can give to all blood types. And we call that the universal donor. So if you look at that, what's the one blood that every one of these blood types can receive? And hopefully you noticed O negative. Since O negative doesn't have any antigens, you can't possibly give anybody something new. You can't give them an antigen that's foreign to them because they don't have any antigens. So O negative blood type is the universal donor. It means you can give O negative blood to everyone. Okay, and so if you have O negative blood, a lot of times that's a very desirable blood type for blood donations because you can give that blood to everyone. Now look at O positive. Why isn't that the universal donor? I can give O positive to anyone with positive blood, but you can't give it to the negatives. The Rh antigen is foreign. Let's look at this. Which blood type is a universal recipient? Which type of blood can receive any type of blood? And that means which type of blood has all 3 of these antigens on it? That would be this. I can give every one of these blood types to AB positive and it would be safe because they already have, this one has A and B proteins. I already have that. This one has B and Rh proteins. Oh, this guy's already has that. No, it's not foreign. This has B proteins. Oh, you can take that too. You already have B proteins. So see how AB positive would be the universal recipient. So if you have AB positive, you can receive any blood type safely. Like I said, we're going to talk a little bit more about this in lab, so you'll get a little more practice. The final thing I just wanted you to notice is this is 6.6. I didn't put the title there, but it should be summarize how the cardiovascular system interacts with other body systems to maintain homeostasis. And again, this is just to kind of get this in your mind, like what do some of our other body systems do, and how does the cardiovascular system interact with those? And you can look this over. This is in your book on the cardiovascular system interacts with the digestive system because it takes nutrients from the digestive tract, delivers it to the cells. It interacts with the respiratory system because it takes oxygen from the lungs and carries it to the rest of the body. There's all sorts of different things. Endocrine system, the blood vessels transport the hormones. The nervous system, nerves help regulate the contraction of the heart and the contraction and dilation of the blood vessels. So the nervous system and the cardiovascular system interact. So I'm not going to ask you a lot about this, but I just want you to thinking about that, of how all the body systems kind of work together to maintain homeostasis.